So hi everyone, welcome to Raaonline. This is Dr. Srivatsan, your faculty for biochemistry, and today's topic is regarding polymerase chain reaction, otherwise called as PCR. This is a very frequent term which has been used nowadays, especially after the outbreak of COVID. Okay, you would have heard this term RT-qPCR, real-time quantitative PCR. Okay, for confirming this SARS-CoV-2 virus. In today's session, we will be seeing exactly what happens in polymerase chain reaction. So, what is polymerase chain reaction first? Any idea? This is nothing but a in vitro replication. This is an in vitro replication. In vitro replication means what? You are trying to replicate inside an instrument, that is not inside a cell. This is done inside an instrument or a test tube. Okay, so this is an in vitro replication process, guys. Unlike the recombinant DNA technology, where the same kind of an amplification of DNA occur inside a cell of E. coli, whereas in case of polymerase chain reaction, this is an in vitro replication wherein the replication is being happening outside the cell, that is in a lab. So, this is in vitro replication of specific fragments of DNA. Specific sets of DNA, that is the DNA which you want to amplify. Okay, so this is just an in vitro replication of specific set of DNA, replication or otherwise called as amplification. And please remember, you are not going to amplify the entire genome, entire human chromosome. We are going to just amplify only the chromosome which is of our interest. So, this is what is all about polymerase chain reaction. So, now we will see what are the requirements for polymerase chain reaction. Okay. So, basically what you are going to do, it is something like a process similar to that of a replication of DNA molecule. So, replication means what it requires first? It will be requiring polymerases. In this case, what are we synthesizing? We are synthesizing DNA. So, what you require? You require DNA polymerases. This will be your first requirement. So, after DNA polymerases, how this DNA polymerases actually work? DNA polymerases need the 3' OH region of a primer, of a RNA primer. Okay, this primer is required, and from that 3' hydroxy region, this DNA polymerases actually starts building up the new strand. Okay, so what you require now? You will be requiring primers, RNA primers. Okay, and what will be the third thing which is required? The third thing which will be required is your buffer and catalyst. What is the third thing which is required? Which is your buffers and catalyst. For what this buffer is required? This buffer is required to maintain your pH for the whole process to occur. It will maintain the pH. The optimal pH will be maintained. The catalyst will be usually your magnesium and manganese. Magnesium or manganese. Magnesium ions or manganese ions. This will be your catalyst for the formation of a new DNA strand. And where all this kind of reactions will occur? All these reactions will occur where? Inside a instrument called as a thermocycler. So, why it is called thermocycler? Because this thermocycler will actually alternate upon different temperatures. This will work on different temperatures. It will change its temperature from high to low at different cycles of replications of DNA. Okay. So, this is why this is called as a thermocycle. Now, please remember what are the requirements now we have seen. The first requirement is your DNA polymerases. Okay. The second requirement will be your primer. The other requirements will be your catalyst, okay, which is usually your magnesium ions or manganese ions, and buffers, which is used for maintaining your pH. And all these things will be loaded in an instrument where this polymerase chain reaction will take place. This instrument is called as a thermocycler. Why it is called as a thermocycler? Because it alternates between different kinds of a temperature for various kinds of a cycle of this replication to occur. So, these are the main requirements for your polymerase chain reaction. Okay. So, now we will see the steps how this polymerase chain reaction will be taking place. Okay. The first and the foremost step is choosing your primer. This is your first important step. Okay. Why this step is extremely important? Because we are not going to replicate the entire genomic sequence. We are going to replicate only the genomic sequence which is of our interest. Okay. For this to happen, we need to select which kind of a part we need to amplify.